So I'm a geneticist working on a problem called animal development. We are interested in a question concerning how organ size is controlled. And to approach this question, we use fruit fly as a model system. One of the luxury of working with fruit fly is that when you discover a new gene, you have the right to name it. For example, we discovered a gene called the hippo because when you mutate this gene, the organ become too big, reminiscent of a hippo. And conversely, we also discover genes which work in the opposite direction. For example, we discovered a gene called Yorkie. When you remove this gene from the fruit fly, the organs become smaller. It's kind of reminiscent of Yorkshire Terrier. Important realization in the last maybe 20 years is that many genes that are discovered that play an important role in fruit fly development or other physiological processes have turned out to be very important and playing a very similar role in humans. So the way I look at fly is basically I think about them as humans with two wings. And when the studies from fly really guide us to look into how the counterpart of these proteins or these pathways play their biological function in humans. And so it's actually, for example, the genes I just talked about, hippo and Yorkie, and their counterpart in human actually play a very similar role. For example, if you remove the human counterpart of a hippo gene from an organ, the organ will grow bigger. And actually they actually go on to form cancer. And so by working on fly, you can make a very precise theory and a prediction about their gene function in human. And then you can do very targeted experiment to test those hypotheses.